Hello and welcome back all. Let's take a look at some neuron physiology, and to do so, let's review from Chapter 3 about the nature of cell membranes. Now you might remember that cell membranes are composed of a lipid bilayer, and the lipid of the membrane is a phospholipid, which has a hydrophobic tail and a hydrophilic head. In an aqueous environment, which is the environment that cells are in, the phospholipids will tend to arrange themselves such that all of the hydrophilic portions migrate together and all of the hydrophobic portions tend to migrate together. This arrangement of the lipid bilayer places the hydrophilic heads facing the extra and intracellular spaces, that is the aqueous environment, and the hydrophobic tails are sandwiched in the middle. Now, if our cell membranes consisted solely of these phospholipids, movement through the membrane would be driven entirely by simple diffusion. Let's pretend for a moment that that is the case, and if so, we would find negatively and positively charged particles in the extracellular and intracellular environment, and although these charged particles might be attracted to the heads of the phospholipids, they would be repelled by the tails that are on the interior of the bilayer. On the other hand, lipid-soluble molecules will diffuse easily through the membrane and will follow their concentration gradients. Within the interior cells are large negatively charged proteins that are unable to move across the membrane. These proteins will play a big role in the establishment of the resting membrane potential, which we will be talking about shortly. Let's now look at some definitions of some key terms that will help us better understand membrane potentials. Let's take a look at the ions that play a major role in resting membrane potentials. The concentration of sodium ions tend to be much higher on the outside of the cell, while the concentration of potassium ions tend to be much higher on the inside of the cell. There are anions in both the intracellular and extracellular environments, but they tend to have a minimal effect, with the exception of the large proteins within the cell that are negatively charged. We know that cell membranes are not made up solely of phospholipids, and we also know that membranes are polarized or charged. We will now look at exactly what it is that causes the polarization of cell membranes. Now, all cell membranes have a slight voltage or potential difference across their tiny expanse. And just inside the membrane, it is slightly negative, while just outside it is slightly positive. And this is caused by the selective diffusion of ions across the membrane. We have to go back to Chapter 3 to review the types of membrane proteins, and the specific membrane protein that is of particular significance is the transmembrane protein called a channel protein. Channel proteins are specific in that they allow only one type of particle to diffuse through them. The channel proteins that are of interest to us are the potassium and the sodium channels, and these channels allow charged ions to move along their concentration gradient through facilitated diffusion. And the concentration of potassium on the inside of the cell tends to be higher than the outside, and the potassium channels tend to be leaky, which means that potassium will be allowed to leave the cell. Negative ions usually have a tendency to follow the positive ions. However, the predominant anions within the cell are the negatively charged proteins, which are too large to diffuse out. So as potassium leaks out, this causes a slight negative charge just inside the membrane. The concentration of sodium is much higher on the outside of the cell, and it tends to diffuse in. However, the sodium channels are not as leaky as the potassium channels, so less sodium will diffuse in than potassium diffusing out. And this again sets up a negative charge on the inside of the cell. The cell membrane tends to be about 60 times leakier to potassium than it is to sodium. These are the main factors contributing to the establishment of the resting membrane potential. However, there needs to be some way to restore and maintain the concentration gradients, and that is the transmembrane carrier protein that we learned about called the sodium-potassium pump. Recall that the pump exchanges 3 sodium out for every 2 potassium in. The pump maintains the concentration gradients that drives the diffusion that sets up the potential across the membrane. We're going to finish up this video by watching an animation from your textbook regarding resting membrane potentials. All right, take it away, Molly.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.